Hi, I'm Rory Cunningham. I'm a final year PhD student in the Department of Nutrition and Exercise Physiology under the supervision of Dr. Scott Rector. So our lab studies non-alcoholic fatty liver disease, or NAFLD for short. And this is a spectrum of liver injury that can range from simple fat accumulation in a liver cell, and if left untreated, this can cause inflammation in that cell as well as in the whole liver, and then the end stage would be cirrhosis, or a lot of scarring in the liver. So one of the mechanisms behind NAFLD development is a dysfunction of the mitochondria, in the liver mitochondria. So mitochondria, as everyone knows, are the powerhouse of the cell, and two of their main roles are to create energy and to burn fat in that cell. So dysfunction in the mitochondria causes a fat buildup in a liver cell. So the reasons behind mitochondrial dysfunction in the liver aren't known that well, and one potential mechanism is a reduction in endothelial nitric oxide synthase, or eNOS for short, as this protein has been shown to keep mitochondria healthy in other tissues. So to figure out whether eNOS is important for NAFLD development, we created a mouse model that was lacking liver-specific eNOS. So in their liver cells only, eNOS wasn't there, but it was present in every other cell. So compared to normal mice, mice lacking liver eNOS actually had more fat accumulation in the liver, as well as a decreased ability of their mitochondria to burn fat, and they overall had more dysfunctional mitochondria. So since we know exercise is an excellent way to make the mitochondria function better and make it more healthy in general, we gave these mice who were lacking eNOS in their liver wheels to run on for 10 weeks to see if this would overcome some of their dysfunctional mitochondria due to their lack of eNOS. So interestingly, the mice lacking liver eNOS did not see the same exercise-induced mitochondrial improvements that the normal mice had. So overall, the reason that the mice lacking, from both studies, the reason the mice lacking eNOS had poor functioning mitochondria was because they had a worse or an impaired ability to remove dysfunctional old mitochondria that were hanging around. So this is a process called mitophagy. So it's where old dysfunctional mitochondria are removed and recycled, so new mitochondria can take its place. So taken together, it looks like mice who were lacking eNOS in the liver cell had reduced ability to create and maintain a healthy mitochondrial pool in that cell, where old dysfunctional mitochondria were still hanging around and not being degraded, and this was reducing the cell's ability to burn fat and causing overall dysfunction in that cell. So because it's still not really known how liver mitochondria become dysfunctional during NAFLD development, these studies are important to try and map out the molecular mechanisms of NAFLD development and to potentially create new therapeutic targets.